Okay, so we're going to move along to our next speaker, and it's my pleasure to introduce Dr. Neil Cashman. Now, Dr. Cashman started his work on ALS back in Chicago many years ago, not that long ago, but many years ago, and actually has been the head of the ALS clinic and the lead neurologist in Montreal at the Montreal Neurological Institute in McGill, then moved to Toronto and had the same role, and is now a co-director and neurologist at the clinic at Vancouver Coastal Health at the University of British Columbia for a number of years. He's also a professor at UBC in the Department of Neurology, and has been a long proponent of how misfolded proteins can act in a prion-like mechanism and can propagate further misfolding of proteins. And we're really fortunate to have him here today to speak with us about his work. So welcome, Dr. Cashman. Oh, we can't hear him just right now. Oh, I think I just heard. Hello?
Can you hear me now?
Oh,
hello?
We can hear you, but it's just a little bit quiet. Is there?
Okay, I can turn it up. Is that better?
That sounds better, I think.
Okay. I'm trying now to actually get the presentation going. I know that I had a little tutorial from Colleen this morning, but can I have it up? But I'm not sure it's on your screens.
No, on our screen we see a list of webinars, and we see, I think, a desktop, but I think you can switch. Are you running two screens?
I have multiple apps open, but
okay.
I do have the slide presentation, which is on top. So
okay. What we had yesterday was someone. There we go. I think if you start it again, it might show up.
Okay. Are you seeing it?
We are seeing it, yeah. So if you start the presentation, I think it will. Perfect, thank you.
Okay.
Thank you. The floor is yours. Thank you.
Thank you. So I will, so I'm also trying to minimize, oh, brother. Are you still seeing my presentation?
No, now we're back to a different presentation, but we'll, maybe if you open it again. There we go, we see it now.
Okay. Is there any way to minimize the GoToWebinar?
No, yeah, what you can do is there's a little orange arrow you should have, and if you click it, so you're going to have to start your presentation up again for us to see it, and then on the GoToWebinar at the side, there should be a little orange arrow, and if you click it, it will actually, oh, you don't have the orange arrow, Colleen?
I did click it, but then it went to another presentation, but I'm seeing my presentation now. Are you?
Yeah. Yes, we are.
Okay, good. My apologies to you, David, and to Colleen, who instructed me properly. So I'm going to talk today about our studies in propagated protein misfolding, focusing mostly on superoxide dismutase 1, but bringing in some of the other proteins that have been studied in my lab and many other labs over the years. I just want to mention that I can disclose many industrial engagements, but none of what I'm going to show you is directly related to any of these companies or licenses. So I want to start with just a kind of general walkthrough of the prion hypothesis, and I know this is probably going back to a kind of biology 101 level, but I think it's important to define our terms. So when I start talking about applying prion-like mechanisms to ALS, we'll have a a better idea. Of course, the prion hypothesis was developed to explain the infectious agent of the prion diseases, and that includes some horrible diseases like Jakob-Creutzfeldt disease in humans, and mad cow disease and scrapie in animals. But the molecular basis of these diseases is all the same. In all of these species, there's a normal prion protein, which is rich in alpha helical content. It's called PRPC for cellular. This protein can be misfolded into an abnormal form. And for the prion protein, that is a beta sheet-rich protein. But for some other prion diseases, it's not the case, prion-like diseases. Anyway, the thing that makes this remarkable kind of epic setting in biology is the ability of the misfolded prion protein to recruit more conformational copies of itself from the normal prion protein. So when PRP scrapie comes in physical contact with PRPC, the PRPC is converted through a still unknown process into more of PRP scrapie. This is an idea that won Stanley Prusiner the Nobel Prize back in 1997 for the original infectious prion diseases. But this idea kind of saturated my imagination when I was first seeing ALS patients in the clinic at University of Chicago, and then on to McGill, and then on to University of Toronto, and finally here at UBC. And the thing that kind of captures the prion idea best is this kind of propagation from an initial site, a systematic spread through upper and lower motor neurons and their tracts in ALS, which of course can be explained by other hypotheses, but fits very well with a prion-like hypothesis. As time has gone on, and there's been this marvelous harvest of mutant genes in ALS, the other factors that make ALS potentially prion-like have emerged. For example, protein aggregation, supersaturation of at least SOD1, the first gene found mutant, the mutations disrupting the protein structure, protein aggregates being toxic to neural cells. I mean, this is the complete shopping list of what one might want for a prion-like disease. Basically, the feature that is least like a prion disease is that SOD1 was thought to be aggregated, accumulated, toxic in SOD1 familial ALS, in which there's a mutation in SOD1, the SOD1 gene itself, whereas real prion diseases, or diseases like Creutzfeldt-Jakob disease, the sporadic form is involved with aggregation of wild-type prion protein. So we came upon this, test of the hypothesis, and it took us years to develop the informative toolbox to be able to ask and answer these questions. Here's a paper published in 2011 in which we've expressed in HEX cells, which is a embryonic kidney, human embryonic kidney cell line. We've expressed a normal mutant, or a mutant giving rise to familial prion disease, familial ALS, called G127X. We've expressed that in these normal human cells, and we've asked the question if the very presence of that protein is enough to trigger the misfolding of wild-type SOD1, human wild-type SOD1. And here you can show we kind of used a molecular trick because this native G127X mutation lacks the entire C terminus. There's a deletion in this region. We can detect G127X using antibodies against a non-native epitope tag, but we also had tools to detect misfolded wild-type SOD1, which would not cross-react with G127X. So you can see in the same cells, there's mutant G127X expressed, and in those same cells, there is misfolding of human wild-type SOD1. This, of course, has many validations in the paper, but one of the most amazing things about this, it's still amazing to me after 6 years, is that we found this conversion process was basically due to a single amino acid, which was tryptophan 32. And David Taylor, you're well aware that you implicated this residue in SOD1 in your PhD studies, in which you found that the presence of this tryptophan at position 32 was associated with the toxicity of a mutant SOD1. But what we were able to find is that it is responsible also for the ability of SOD1 to confer its abnormal conformation in molecule after molecule after molecule. This is unlike mouse SOD1 that lacks this residue at position 32, lacks the tryptophan, but in human SOD1 conversion, conformational conversion, the tryptophan is required in both the misfolder and the misfoldee. We were able to demonstrate that, much to my surprise. So this is very intriguing because that tryptophan can be drugged. It's a surface tryptophan, which is unusual because usually tryptophan is buried, but there are molecules discovered by Wright et al. In 2013, including an approved anti-cancer drug called 5-fluorouridine that binds tightly to this site at the surface of SOD1. So we tested the irresistible hypothesis as to whether 5-fluorouridine could inhibit the propagated protein misfolding of SOD1, and found, much to our gratitude, that that yes, indeed, low concentrations at the level of less than a micromolar potently inhibited this propagated protein misfolding, which was mediated by tryptophan. So the molecule-to-molecule conversion, of course, that's prion-like, but in order to demonstrate more prion-like phenotypic characteristics, we developed an assay to transmit wild-type SOD1 misfolding from tissue culture to tissue culture to tissue culture. And what we were able to demonstrate is that once wild-type SOD1 is induced to misfold, that can be propagated across tissue cultures indefinitely. We've only shown 5 passages in this study, but we've taken it up to 20 passages in the lab, and there's no decline in the robustness of this ability to transmit. So this is very prion-like, and as time has gone on, Jake Ayers and David Borschelt have developed an animal model of this transmitted misfolding of SOD1 that results in paralysis of the mouse. So it is highly consistent with the human disease. But what stimulates wild-type SOD1 to misfold and perhaps propagate in sporadic ALS. So we had that question for many years and decided to test whether wild-type SOD1 could be found to be misfolded in not only SOD1 familial ALS. You can see in the upper left-hand corner, there is ample evidence of misfolding and aggregation of mutant SOD1, but also in sporadic ALS with TDP43 deposition, we were able to find evidence of SOD1 misfolding, and also in FUS mutant familial ALS, we were able to find evidence of wild-type SOD1 misfolding. So Eddie Pokrzywski in the lab actually developed a system in which we could express mutant FUS and mutant TDP43 in cells in vitro. These are mouse spinal cord cultures derived from mice that express human wild-type SOD1. Remember, I told you that the mouse SOD1 is kind of inert to this propagation by virtue of lacking the tryptophan. So in every cell in which we saw mutant FUS or mutant TDP43 expressed, there was also misfolded wild-type human SOD1. And Eddie has gone on to demonstrate that this SOD1 is not just misfolded, it acquires the property of transmitting from cell to cell, which of course is, again, very prion-like. And here we're using the wild-type misfolded form, conformational form, to transmit from culture to culture. So we have also found that this transmission is inhibited by 5-fluorouridine, which suggests that even in wild-type ALS or sporadic ALS, if there is evidence of ongoing misfolding, that 5-fluorouridine is a potential lead drug to block this process. Now, I want to just spend a minute or two talking about the transmission between cells. We published back in 2014 that this can be mediated by two mechanisms, one by naked aggregates, which transmit from cell to cell, and the other one through exosomes and other extracellular vesicles like microvesicles. And here again, a study by Eddie Pokrzywski showing that SOD1 familial ALS spinal cord homogenates are able to transfer misfolding and aggregation from one cell to another. This is not seen with the sporadic samples, in which, of course, the SOD1 appears to be, the misfolded SOD1 appears to be in a different form. Interestingly, this aggregate mediated transmission can also be blocked by 5-fluorouridine and also by antibodies directed against misfolded SOD1. Sarah Fernando in the lab has been studying exosomes and microvesicles that have been derived from the brains and spinal cords of mice and humans with ALS. And here we are showing transmission of mouse G127X exosomes to mouse cells that express human SOD1. Again, using this trick that we won't detect misfolded wild-type SOD1 in these cells unless the G127X truncated mutant has the ability to transmit its misfold to wild-type SOD1. So it's a long story, but human mutant and human wild-type SOD1 are true prion proteins, and I have to put that in quotations because there's quite a bit of discussion and argument about the word prion for a disease like ALS or Alzheimer's or Parkinson's by transmitting a misfolded conformation between cells. And Anne Bertolotti has done this, and we've done this. Transmission of human mutant SOD1 and human wild-type SOD1 is neurotoxic, and as I said, Jake Ayers has a wonderful experiment in mice. Darryl Boscow has actually been able to isolate misfolded human wild-type SOD1 from sporadic cases and show that this SOD1 inhibits axonal transport. And we've done experiments showing that wild-type misfolded SOD1 is toxic to neurons and to HEC cells. Clearly, the state of pathological human mutant SOD1 and wild-type SOD1 differs between SOD1 familial and sporadic disease, and we believe that this may be insoluble aggregates versus soluble oligomers. Jake Ayers also believes that, and stay tuned. The primary mode of human mutant SOD1 and human wild-type seeding transmission may differ, aggregates versus extracellular vesicles. Human mutant SOD1 can seed human wild-type SOD1, and SOD1, you know, this is our reporter system in a TDP32 dependent process. And TDP43 can also stimulate wild-type and mutant SOD1 to misfold. Again, this is through a TDP43 TRP-mediated dependent process, which I haven't shown today. So here are the acknowledgements. I'd like very much to thank ALS Canada. I'd like to thank my collaborators at UBC. Steve Plotkin is a computational physics person who works with us. Ian McKenzie has been very supportive in terms of tissue and his knowledge. University of Toronto, Avi Chakrabarty, Christine Van Velde, University of Laval, University of Alberta, and University of Saskatchewan. Much of this work is supported by the groups in the upper left-hand corner, but some, more particularly our Alzheimer's propagated protein misfolding work, is supported by Promise Neurosciences. I thank you for your attention, and I hope that I've left a little bit of time for questions, but Dave will tell me otherwise if that's the case.
Thank you, Dr. Cashman. Well, we have some questions, but I think they're probably lengthy answer questions, and could get pretty deep digging into some of the answer would be a bit long. So I'm going to actually just ask you one quick question, which is that, as you probably know, there are a number of treatments to focus on SOD1 as a potential treatment target for people with familial ALS SOD1 mutations. You've got something like copper ATSM that's actually looking at a small trial in Australia where they're doing actually people who have ALS, including sporadic disease. And I was wondering if you think it would be interesting or worthy to try these types of therapies on sporadic ALS. Your thoughts on that?
Yes. So yes. So the short answer is yes. There is a potential problem in that all of the formative data for this has actually been generated in mouse models, which of course are expressing human mutant SOD1. There is evidence from Avi Chakrabarty and others that wild-type SOD1 and mutant SOD1 unfold differently. So it is possible that copper ATSM might only not work in sporadic ALS, but it could make things worse. So that's that's a painful possibility, and I've discussed this with the people doing the Australian experiment, and I agree with them that the only way to know for sure is to actually try it. And it sounds hard, but yes.
Okay. Well, thank you very much. Maybe I'll be able to forward you some of the questions that we've had from the audience, and we can follow up with that. But thank you so much for your time and for the presentation, and have a great rest of your day.
Thank you.
Thank you.